get started? Yeah.
Sorry, so when do we have again to finish the final?
On Sunday at midnight is the yeah, so you will get an email from Pam with the exam, and then you yeah. Sorry.
You were in the middle of
Oh,
something.
so you will get an email with the questions, and then when you've solved them, you get a document with the questions, and then when you've solved them, you go to this website, you get a link, and you fill in your answers, and then so before midnight on Sunday. Yeah.
So I was just thinking, like, for example, I know that the email address that you have that I'm registered with is my @NYU address, but I've been submitting all my courses and assignments through my NYUMC address. That doesn't happen.
No, either one. Either one. So actually, so for the exam, we try to find all your NYUMC emails, and but for some of you, you might get it. If you don't get it to the NYUMC, it might go to your nyu.edu email. So please check both. But the majority of you will get it to the NYUMC. Good. So, so I'm going to talk a little bit about experimental design today, and this is of course something that you'll all do quite a lot, and and the main thing is that it's easy to make a mistake, and do it in a way so that the results really don't make any sense. So this cartoon is just one example. We're trying to measure one thing, but then there is a side effect that we don't anticipate, so our conclusions are completely wrong. And this will happen quite often, and I mean the main thing with experimental design is to constantly think about what could go wrong, what haven't I thought about, and also when you then get to analyze the data, if you find something really interesting in your data analysis, it quite often is that you've done something wrong, unfortunately. And especially the more interesting your finding is, the more you have to go back and really think it through and do other experiments to test it to make sure that it isn't just something else. So there are some basic experimental design strategies that we'll go through, and the main sort of purpose with these is to focus on two things. One is that there is chance, so we have some kind of randomness always built in. We have to figure out how well we can measure things so that we can overcome chance. And then the other thing is bias, which is that an effect that we really didn't think about that systematically changes the results. And that's and if we have either of these two, can make our conclusions invalid. So one can see an experiment as a process where we have some factors that are controllable and other factors that are uncontrollable. So just, can you give me a few examples of controllable factors in your experiments? Anyone? What would be a controllable factor?
Temperature.
Yes. I mean, so actually temperature could be both a controllable factor and an uncontrollable. In your dish, you can control it usually pretty well, but the room temperature can be quite uncontrollable if you don't have a lot of money to have a good air conditioning system. So as you see, some of these factors are, it's not clear. They can both be controllable and or they can be uncontrollable. And you'll have to think through in your case which factor is in which category. And of course, if you can move some of the uncontrollable factors into the controllable, that's always an advantage. So some of these things are quite sort of obvious, but it's still good to have a sort of a checklist when you design an experiment. So first is to state the problem, what you're trying to test, what's your hypothesis. That is a good point to start. And you probably, that part most people do. But then to go through and select the response variable, what do you want to measure? See what are your controllable factors and what's the range that you can change these controllable factors within. And then try to list all the uncontrollable factors that might affect your experiment. And later on, we're going to go through a few examples where both factors that could have been controlled and uncontrollable factors had very large effects on the experiment. So then choosing an experimental design, so we'll talk more about that. Then actually do the experiment and do the measurements and analyze the data. And after analyzing the data, go back and do the next experiment. And it's always, if possible, best to sort of design smaller experiments and then have do an iterative process to change the experimental design slightly on the basis of the results from the previous experiments. And so that's what's usually recommended. So if we start with just looking at exploring parameter space. So if we have a number of factors and we want to see how they affect the results. So in this case we have k number of factors and we just change them one at a time. So we also just look at two values for each. So the minimum within the range and the maximum range. And so then we need to do 2k experiments to test all of them. So that's, if we don't have too many factors, that's not an enormous number of experiments. But now often these factors that we're testing, they depend on each other. So preferably we want to test all the combinations. So now if we have just two factors, if we test all the combinations of them, we have 4 experiments. And then 3 factors, 8 experiments. So we'll end up having 2 to the factor of k experiments if you want to just, and this is just now 2 settings for each factor, and but we want to do all the combinations. So for example, if we have 10 factors, it ends up being more than 1,000 experiments, which is a lot. So that's not really feasible in a lot of cases. So another thing that we need to think about is randomization. Randomization is mainly the reason we do it is we want to guard against unknown and uncontrolled factors. And we should do the randomization with respect to anything we can think about. So for example, if we do analyze serially a lot of samples, then what we should randomize the order of that the samples are analyzed in. If we have several instruments that we use, then we should also randomize which samples are run on which. And also, if we have different materials, different batches, then we should randomize that. And the reason is just trying to show that here. So in this case, we have two groups. One is the red, and one is the black. And we have, in this case, this is the order that we measure them. And in the case when nothing changes during our measurement, it doesn't really matter if we randomize or not. We still, these two, there is no difference between these two groups. So in both cases, both with the randomized order and the non-randomized order, we get a high p-value. There's no significant difference. So then this is the same case here. But then let's say that the sensitivity changes during our measurement. So slightly we have an increase in sensitivity for some reason during, and we don't randomize. In this case, because of this change that we didn't anticipate, we get a very small p-value. But if we would have randomized, we would have again gotten a high p-value, so no significant difference. So that's the main reason to randomize. So if there is something that changes, and there is often, especially if we do lots of measurements that last for several days. So for example, one thing that is so in proteomics, where I've been working a lot, if you run, it takes quite a while to run each sample, a few hours. So if you have lots of samples that go over several days, one effect that one can see often is that if you don't control for the room temperature during the summer, you see quite a big difference in sensitivity if you run it at night or in the middle of the day. So that's an example of this. So then we have something called blocking, and these are blocking is used for known and controllable factors. So one example of that is what's called a randomized complete block design. So in this case, we have 4 different instruments, and then we have samples, and now we're running each sample once on each instrument, and then we get then for each instrument we randomize the order of the samples, but we make sure that we run each sample on each instrument. Then in the case when we have different instruments and different operators, then we need to also distribute the samples over the instruments and make sure that also each operator analyzes each sample the same number of times and on the different instruments. And so in this case, in the first case, the randomized block design, we just had the restriction on randomization that the order that each sample should be analyzed on each instrument. But here we have also the restriction that each operator should analyze each sample. So now we have restrictions both on the columns and on the rows. So this is a little bit like Sudoku, where you also have different kinds of restrictions. So one can see this as playing a game when you design your experiments. So then replication. So here, the reason why we do replication is that we have variation in the measurement. And if we do many replicates, we can both estimate what the variation is, and we can also make it smaller but there are different types of replicates, and we can do so. The best replicates to do, of course, is the biological replicates, because then we can draw conclusions on the biological variability. But often that might be difficult, so at least we should do technical replicates. But it's definitely important not to think that we can, by just measuring technical replicates, that definitely limits us in what biological conclusions we can draw. Yeah, so a biological replicate would be, for example, that we take tumors from different patients. So that would be biological replicates. A technical replicate would be that we take one tumor and cut it up into different pieces, and or we homogenize it and divide it up, and then analyze each of the piece separately. And then a process replicate could be, for example, if you have a cell culture and you grow the cells, it's the same cell, but you grow them on different plates. So that could be a process replicate. It's not always completely clear what is what, and sometimes, for example, the cell culture case, I've seen people claim that as a biological replicate, but I would argue that it's not.
So the process replicate for you is kind of the in-between to suggest for stuff that's coming from the same cell line that's treated the same way but in separate containers.
Yeah.
That's a process.
Or in separate days. Or,
But it's all coming from the same place. Yeah. That's always been people, and I know a lot of people call those biological.
Yeah. No, I've seen that too. Yeah. And I usually point that out if I don't agree with that. But they don't agree with me always.
So David, like on those lines then, to make it a valid biological replicate would be independent, like different cell lines.
Yeah. So different cell lines. So that's. But I mean, of course, the problem with all this is that it's much easier and cheaper to do technical replicates than it is to do biological replicates.
If you have like a one cell line but a different hairpin, or like a different clone into a different vector, like expression sequence, does that count as a biological replicate then? Because that's biologically different?
Yeah, I mean, usually that would be the original cell line, maybe would be your control, and then you do some manipulation with it, and that would be your experiment.
But I mean, between two, like if I knock down a gene with one hairpin, then if I have another, like, even if it were the same hairpin, like, is the same hairpin cloned into a different, like, lentiviral vector? Is that a biological replicate, or does it have to be a different hairpin? So that would be process replicate, but biological would be a different hairpin in the same cell line. That would count?
No, I mean, it probably would, no, a different cell line in that case, using the same hairpin, because that's your experiment. So you have your original cell line, and then you do some manipulation. And then, but to have biological replicates, you would preferably do this in several cell lines.
Yeah, I don't know, anyway.
I mean, I guess it would be valid if you make it like a stable cell line that's different, right?
Well, that's what I'm asking, because I've had this discussion with like other people before, and there doesn't seem to be a clear answer. So.
No, I mean, I don't. I mean, in this case, there's probably not a clear. I mean, it's some people argue for one thing and some for other. Yeah. So then, we talked about this last time, that if you do a lot of measurements and take the average, then the error of that average decreases if the more you measure. So this is and it decreases as the square root of the number of samples. So you can see it and it's also for a quite wide range of different distributions, this is the case. So we see here, if we have 3 replicates, we have quite a wide distribution, but if we do 100 replicates, it's very narrow, the mean is very narrowly defined. And so this is another reason to do replicates. And as we talked about last time, that even if you have distributions that are far away from normal, if you take the average, and for many replicates, the distribution of the average will be get closer and closer to normal. So this was the central limit theorem. So more replicates are better. So now, I don't know if you can see this well, but, sorry? Oh. So this is a bad experimental design. So this is before and after, samples before and after treatment, or patients before and after treatment. We have sampled them. Here, this is a proteomics experiment. So this is the gradient length of the chromatography, and then the date that the analysis was done. And it was done in 2 laboratories, laboratory 1 and 2. And these are the patient numbers. So for example, patient 6 is here and here, and so on. So for each patient, there should be 1 before and 1 after. So why is this a bad experimental design? And this is actually real. This is one of my collaborators came to me with this.
Your own control factors, so the length of the gradient length, 3 hours and 1 hour, is different between before and after. You have different laboratories that don't seem to be randomized on either side, so they're not balanced for the effects that could be coming from one laboratory to another, which, as you say, could be temperature, 100 things, right? It could be completely different scenarios.
Yes. So that's all. So there are many things that were wrong with this. So everything, all this should have been randomized, and also one shouldn't change the experimental protocol during an experiment like they did here. I think what happened here is that when they were running the samples 3 hours, then they figured, oh, we don't have that much time, let's speed this up. That's completely wrong. But also everything else, so that they should have not changed, but these other things they should have randomized. So this is, this is the worst case I've seen of an experimental design during many years. But it's not unusual. And the thing is that they came to me when they had done the experiment, and they asked me to analyze the data. So I started analyzing the data, and then it didn't look right. So I started asking questions. So they didn't have a table like this. This took me asking them several times before it I could piece this together. So that's a real life example of when things go wrong.
Could you salvage this?
No. I told them to throw all the data away, and they did actually, and they redid the experiment. Unfortunately, not with much better results. Then the experimental design was good, but it didn't end up being anything interesting. So I'm going to show another example from mass spectrometry based proteomics, and I thought I'd give you a little bit of background on proteomics. So what we do in proteomics is that we take samples and then maybe separate the proteins and then at some point digest the proteins into peptides, and usually with trypsin, because trypsin produces peptides that are easy to, relatively easy to separate in chromatography, and they behave well in mass spectrometers. They ionize well and they fragment well and so on. So finally we end up with lots of mass spectra. And so we start with usually a sample with quite a few proteins, maybe sometimes if we do tissues and so on, like 10,000 proteins. And then we cut them into peptides and end up with hundreds of thousands of peptides. So then we do lots of extensive separation of the peptides, and then try to run the mass spectrometer as fast as possible to fragment as many of the peptides as possible. And then we end up with mass spectra. So on the x-axis we have the mass to charge ratio, and then intensity on the y-axis. And we can measure the mass to charge very accurately, down to a few parts per million. And we can use that to identify which peptide is in our sample. And this can be done quite accurately, but it's not like DNA sequencing. We can't just read off the sequence. We look at the fragments. We get sometimes the charge stays on one end of the peptide, sometimes on the other. We have missing data, so we don't get fragmentation between each amino acid in the peptide. We have some peaks are very large, some peaks are very small, depending on the chemistry. So it's a little bit tricky to do. But then we can also quantify based on the intensity of the peaks. This is the quantitation we can't do as accurately. But we can, and we usually need some kind of heavy isotopes to do an internal standard so that we can compare different samples. But in general, what we get out of the mass spectrometry is identified peptides and their quantities. So then, when we have these peptides, then we roll them up into proteins. And now we have the problem is that we don't see all the peptides for a given protein. So we usually see some peptides are too small or too large or too hydrophobic, so they're not seen. We have the problem, of course, that often we don't have enough information to distinguish different isoforms of the proteins. But what we get in the end is quantitation for a lot of proteins. So this is just an example of where we've done measured two samples. So in this case, we don't have replicates, so we measured one sample once and the other sample once. So now we have a quantitative measure of how much of the protein is in the sample. So in this case, it's an indirect measure called the spectrum count, but it does work reasonably well when we look at the same protein in different samples, if we have, not for very low abundant proteins, but for reasonably high. So you see that there is a correlation here, but at low amounts, we don't have a very good correlation. And then we have actually here, so this is log 2. If you have a spectrum count of 1, that would end up here at, if you take the log 2 of it, at 0. We've also put arbitrarily placed the ones that are only present in one sample at the small negative value here. So these here are only present in sample 2, but not in sample 1. So then we can turn this around so that it's a little bit easier to look at it. So now we instead plot the log ratio of sample 2 divided by sample 1 here, and the average intensity, or the sum of the intensities on the y-axis. So what we see here is that if we have very high values, high intensities, the ratios are much tighter, there's much less of a spread, and then down here we have much more of a spread. So of course what we're interested in here is the proteins that are outliers. So for example, these proteins here and these here look like they would be different between the two cases. So one thing we can, since we don't have any replicates, what we can do is base our outlier analysis on the overall distribution, assuming that most of the proteins don't change between the samples. So we can, for example, look at these three different areas and see that up here we get quite a nice curve that is slightly skewed, but quite symmetrical and close to normal. And then as we go down in intensity, it becomes wider and wider. So we can use this and then say that, okay, the ones that are out here are pretty far away from what what the majority is, so we can say that those are different between the samples. Yeah.
This is just my question, but do you use the raw data for sample to over sample 1 ratio, or is it after you normalize the two counts?
Yeah, so we haven't, so in this case we haven't done any normalization, so that's another thing. So usually we would also normalize this, but in this case we've just taken the raw count, actually.
Then how do you know whether you injected or analyzed the same?
Yeah, I mean, you see that if you don't actually, so you see it's slightly off. It's not exactly centered at 0 here. So you see that the whole thing moves in one or the other direction. And it has, I mean, it is more, I don't know if that's easy to see, but I should have put the line at 0, but I think it's slightly moved to the left. Not a whole lot, but it's definitely, it's not, and that's quite often the case, that you try to inject the same amount, but it's not always possible.
So that line gives you sort of like rough idea whether you inject the
Yeah, yeah. And then you use that to normalize also. But in this case, it's not normalized, but it's still pretty good. Yeah. So what we can do is we can argue that this could be that these outliers are different, but we don't really know because we don't have replicates. So if the same sample we actually did run in 3 replicates, and then we see, so the red points here are the ones that are significantly different after multiple testing correction also. So we see that most of the outliers are actually different, but we do see like this one is pretty far out, but it's not significant. We have one here, we have these 2 here. So it's really, I mean, the conclusion is that one should do replicates to do proper statistics. Because really the first arguments that I did with these outliers is really not valid in general. So we talked a little bit about this, about that if we really want to establish a measurement, we can only do this for, of course, a few peptides and proteins, but we really need to establish a calibration curve where we titrate the amount and see where we get the lower limit of quantitation, and show how well we can do the linearity, and if we have systematic errors like here, that we do measure quite precisely what we can reproducibly measure the same thing, but it's off from the actual value. If you only have a few things that you want to quantify, you should definitely try to establish these calibration curves. So then, a little bit about multiple hypothesis testing. So there are two questions written here. So first one, we're pointing to a specific protein and asking if there is a difference between two samples. And in the second case, we're asking, are there any proteins that are different between the two samples? And so implicitly in the second question, we're asking separately for each protein that we're measuring, is the first protein different? Is the second protein different? And so on. So then we can do some kind of statistical test to get a p-value on the first case, just on the measurement on that. And we can, for the second question, we can calculate the p-value for each, but we then need to correct for that we've performed many tests. So the one way of looking at the multiple testing is that, for example, in the New York lottery, if you buy a ticket, the chance of winning is really small. But yet, every week, someone wins. So that's sort of the thing that the second question, where you ask an open-ended question, is there a protein that's different between the two samples? So that's the same thing. So one way of doing this is called the Bonferroni correction, which is a very simple correction. You just multiply the p-value by the number of tests you performed. Usually for high throughput assays, this is a very conservative correction, and you'll end up usually with nothing that's significant if you apply it. But it's easy to do, so we can definitely do it. The Bonferroni correction is very useful if you maybe do 2 or 3 tests. So often in clinical medicine, you test a few different parameters, clinical parameters. Then it's often used, but it's only for a handful, maybe, of tests. So then, yeah.
So when is it more appropriate to do that versus doing an ANOVA and then using the post-hoc test to get corrected?
Yeah, so that's, I mean, it's independent. How you calculate the p-value is, I mean, doesn't matter. So you can use, depending on experimental, then you can use ANOVA, t-test, or or if you have a correlation, you can calculate the p-value from that. Then the correction for multiple testing is a separate thing for that.
So are you suggesting then that you would correct for multiple comparisons even within an ANOVA? So if you have an ANOVA that shows you that there's a significant difference between 2 proteins, let's say, if you run a multi ANOVA, would you then still Bonferroni correct based on the number of proteins that you're running?
Yeah, I mean, if you, yes. Yeah.
So that's what you're saying, where it's like the p-value would still be correct for multiple comparisons.
Yes.
If you're using multiple t-tests
or multiple tests of any, including ANOVA. Yeah. So it's just doesn't matter what the test is. It's just that you apply it many times. So just to illustrate this a little further, if you just simulate you sample from a normal distribution with, in this case, a sample size of 10, and then you do this and you do 2 groups every time of 10, and you just do a t-test, and then you repeat this 1,000 times, and you plot a histogram over the distribution of the p-values over between 0 and 1. And it's if you do this very many times, it's pretty much a uniform, should be a uniform distribution. But of course, we have some noise in this case. So as you see, you end up having the average. So now the bin size is chosen so that we have 20 bins between 0 and 1. So the size of each is 0.05. So this first bin would be where we typically say that things are significant, less than 5%. So in this case, of course, since we have this uniform distribution out of 1,000 tests, in this case a little bit more than 40, but on average it should be around 50. So 1,000 divided by 20. And if we see that if we go to 10,000 tests, it's close to 500, and if 100 tests, it's in this case 4, but on average it would be 5. So this is now another explanation for why we need to do the multiple testing correction, because if we just keep the 5% cutoff, it would we would at random ending up with a certain number depending on the test we do. Okay, so now this was completely random data, so we didn't have any true positives. But let's say that now we have out of the 1,000 tests, we have 30 that are actually where the means of the distributions are different, and in this case very different. So they will all end up in this column here, and but what we'll see is that we have 60 here that are at random, and then we have plus these 30 that are non-random. So now in this case it ended up being a little bit more than 90, and the reason is that these are separate simulations, so we don't actually know. So the random simulation was a different one. So but what you see is that in this case a large fraction of the ones that would be considered significant if we had a 5% cutoff will be false. And then if we have fewer tests, there are a larger fraction that's true. But if we have many more tests, like 10,000 tests, there's almost nothing of these that are true. So what we can define is then instead a false discovery rate. And we can plot this false discovery rate. So this is as a function of p-value. So at low p-values, we have a low false discovery rate, and then it increases and approaches 1. And then, of course, it increases much faster if we have many tests. So this is why we then do the correction. So we decide that we want to choose a p-value cutoff that corresponds to, let's say, 5% false discovery rate, or 1%, depending on your application. And so in addition to false discovery rate, if we put the p-value very low, of course, we won't get any false discovery, but we won't get any of the real ones either, because as we decrease the p-value, the false negative rate goes up to 1. So for example, if you have a small difference between the means, then and we set the p-value really low, we don't have any we still have quite high false discovery rate, but we don't have any of the real ones that we find. So this is just a little bit more background on why you should do the multiple testing correction. So then another thing we sometimes need to decide is, let's say we want to determine the area of a peak. So in this case, it's we have time on the x-axis, and we need to decide how many points should we sample to get a good estimate of the area. So let's say we want to get an estimate that's better than within plus minus 10% of the area. So who wants to guess how many samples we need over the peak? You can just guess. Give me a number.
80.
8. Anyone else?
42.
Okay. One more.
100.
Okay. Yeah, I mean, it does get better the more points you have. That's true, but it's actually enough with 4 to get better than 90%. So if we see here, so 4 points we get almost always better than 90%, and then it improves. So it's actually surprisingly little, but it's an important thing to know. So if you need to, you sometimes need to make a trade-off between measuring different things within a finite time, and then it's good to remember that you don't need that many points to if you don't need very good accuracy.
So is it 90%? What does that 90% actually mean? Is that you are able to predict with 90% certainty the Gaussian distribution?
Yeah. So it's 90% of the points will be within this range. So in this case, it's maybe 0.94 and 0.99. So sometimes you have outliers, but most of 90% of the time you are within this range.
So why is it then that if you're testing for a normal distribution on a data set that's less than 30, do you see a large number, or a large time when you're testing for normality, you don't actually, it doesn't actually qualify? So if you were to put for a normal distribution,
yeah. So I mean,
data points, the odds of it coming back to you saying it's normally distributed.
This it won't tell us. We just assume the normal distribution. So it doesn't tell us anything about whether it's normal. It just this is how accurately we can estimate the area under the curve.
Okay.
So that would be a very different thing. So now, finally, a little bit about molecular signatures. So one thing that we often want to do in medicine is be able to predict from taking a sample and then doing different kinds of predictions. So one would be to, for example, in this case, determine whether it's it's ALL or AML, so two different types of leukemia, and so we see that if we have this about 50 genes maybe, we can very accurately separate the different cases. But also, as you see, there's not no one gene can perfectly separate the two groups, but together we can, with many genes, we can do that. And this is something that is very popular nowadays. I mean, people are trying to develop these, what the FDA calls them in vitro diagnostic multivariate assays. So you measure many genes at the same time and try to predict, try to make them as biomarker for something. So you can try to use this for, develop this for many things. One is diagnosis. but also prognosis. So you take a test and you try to predict how the disease will progress. But probably most interestingly for precision medicine or personalized treatment, so to select which drug to use and using it for dosage and so on. So this is definitely something that people are trying to do a lot now. And sometimes it's very successful. So this is just one example, Oncotype DX. So here, and most of the successful ones are for very specific use. So this is a signature of 21 genes, and it is used to predict what's the risk of someone with ER-positive breast cancer, what's the risk that they relapse. And that is, and it has been used now for many patients, and people are willing to pay quite a lot for this. But so the main thing with it is that for these tests is that one has to have a very precise clinical question and not for each of these tests. Those are the most successful ones. Because the general tests for screening, as you probably read in the newspapers, are not, even though things that have been used for decades can be questions like in some countries mammography is no longer, I think in Switzerland they don't recommend it anymore, for example. So if you can have a much more specific clinical question, it's usually more successful. So back to proteomics, which in 2002 there was a paper in Lancet where with using a proteomics test they saw very good results. So here this is to detect early detection of ovarian cancer from a blood sample, and they reported 100% sensitivity and 95% specificity. This was quite remarkable because you can imagine early detection of any tumor in the blood. You have a small tumor, some proteins leak out into the blood. There is a very big dilution. So you need a test that really can detect very low amounts of protein. But in this case, it was all wrong. And people figured it out after a little while. Luckily, they actually made their data available so people could reanalyze it. And then it was reanalyzed, and it's not completely clear what happened, but they definitely in the lab treated the samples from the ovarian cancer patients. They ran them in 1 week, and the next week they ran the controls. It's not exactly sure that that's what happened, but that's what the conclusion was. So it was there was a company that got lots of money to develop the test from investors, but it failed FDA approval, and in the end it was actually very damaging in general for the proteomic field. So here they developed an algorithm that looked at a pattern. So in this case, it was mass spectrometry, but they looked at high abundance proteins and just saw a pattern. So they didn't know what each peak meant, but they did developed an algorithm to separate these. But then in the end, it wasn't reproducible. And both of the main people actually lost their jobs. But they did find other jobs. so for a molecular signature to work well, we need a well-defined clinical problem, and a lot of people fail on that and just take some samples of convenience that their friend can give them and try to develop some biomarkers that don't mean anything. But people are now more and more trying to do better. Then we need the computational analysis and biostatistics that you've learned in this course. I mean, it might be, if you really want to do this, you might take a few more courses on that. But and then usually some kind of high throughput assay that gives you a lot of measurements to try to build your marker from. So another thing that is actually very tricky is to choose the right normal group. So if you're looking at a certain disease, and what is your normal, what do you compare to? And so at least at basics, one should make sure that everything, so people's age, gender, and so on matches. So, but as many of these overall clinical parameters should match. And in this case, it's two different drugs for breast cancer that were tested, and the researcher made sure that all of these clinical parameters were very similar. And so if you are interested in this, David Ransohoff's paper, and he wrote several papers, including this one, Bias as a Threat to the Validity of Molecular Marker Research. And bias could be a lot of things. I mean, simple things that, let's say you collect your normal in one clinic and your patient blood in another clinic. Or that you, so there could be a lot of things like that. And this is just some more how to address this bias. Okay, so to summarize this, we talked a little bit about chance and about bias, that this could be a problem for the conclusions. We talked a little bit about controllable and uncontrollable factors that affect your experiment. We then mentioned randomization, which is definitely something you need to do to make sure that there are no unknown and uncontrollable factors that will affect your conclusions. The other thing was replication, both to know what the errors of our measurements are, and also to minimize them. And then we talked about blocking to control for known and controllable factors. And also multiple testing is very important to apply if you do many statistical tests. And then we talked a little bit about how this applies to molecular markers. So the main, I mean, additionally, we should think. That's important. It's not, you can go through and do these formal experimental designs, but you should also think, really use your domain expertise and think about the problems, of course. Don't design too complex experiments. It's much better if you can, at least to keep it simple. And then it's best to do iterative experiments. And also, one thing that we haven't touched upon is this difference between statistical significance and practical significance. So the statistical significance, what we do is we calculate a p-value, and then if the p-value is low, then we can we say that there is a difference. But if this difference is very small, so that it doesn't have any practical consequence, it's really not very interesting either. Okay, any questions?